Last presenter this morning will be Dr. Richard Farrell, who will be speaking to us about the topic of nutrition and rotations. Dr. Farrell is an associate professor in the Department of Soil Science at the University of Saskatchewan. He's also the Saskatchewan Ministry of Agricultural Research Chair in Soils and Environment. Richard's research focuses on nutrient cycling and environmental agronomy, including the effects of agricultural practices on soil quality and greenhouse gas mitigation in agroecosystems. Dr. Farrell is also coordinator of the Prairie Environmental Agronomy Laboratory, which supports research aimed at developing strategies that enhance or maintain agroecosystem capacity and integrity. This work, in turn, enhances the environmental and the economic sustainability of soil resources and establishes a low GHG footprint of prairie cropping systems. Please join me in welcoming Richard to the podium.
Thank you. So I'm going to talk a little bit about the work that we're doing on crop rotations and how it sort of fits into the carbon theme of the meetings today. And what we're doing is looking at sort of the other side of carbon fixation, which is carbon doesn't sort of exist in the environment by itself. So nitrogen is a big part of this, and people have talked a little bit about it. Daniel has, Brian mentioned it about nitrous oxide. That's one of the things that we're looking at. And so we look at using how can you sort of use crop rotations and what's their role in sort of managing nitrogen losses so that what you're doing is optimizing those net carbon gains. So this is the other side, is if a system is going to be a net carbon sink, you have to look at the amount of carbon that's coming in. So the plants are photosynthesizing, taking in carbon dioxide, and then converting it into root material and shoot material, it goes back in as residue, converted to organic carbon, and stays there. But at the same time, plants and microorganisms in the soil respire carbon dioxide. They either consume or produce methane, and they produce nitrous oxide. And so whether it's a sink or a source altogether as the system depends upon what the balance is between all of those things. And so, we talk about carbon sequestration. One of the things you want to do is get more carbon into the ground. And an easy way to do that is grow more plants, get bigger biomass, put more residue into the ground. The problem with that is in order to do that, you need to put more nitrogen on. You add more nitrogen, you get more nitrous oxide. It's about 300 times more potent a greenhouse gas than CO2 is. So you've got this balance going on. Are you putting in more carbon or taking out more nitrogen? And so that's what all of this research is sort of aimed at. And this is research that I'm doing with Diane Knight, who's an agronomist in the Soil Science Department at the U of S, and Reynold Lemke, who's a research scientist with Ag Canada at the Saskatoon Research Station. And we have a postdoctoral fellow, Li Ting Liu, who's working on this as well. And so I'll be talking about some of the work that we're doing as a group here. So the first thing is just to sort of go over a little bit of this is the greenhouse gas basics here, sort of. The three main gases for agricultural concerns are carbon dioxide, methane, and nitrous oxide. And you can see that it has these different global warming potentials. Carbon dioxide set at 1, methane is at 25, nitrous oxide is 298. And what you can see, the graph here is this is the percentage of the ag sector greenhouse gases in Canada. So, we're getting about 8% of the total CO2 emissions from agriculture, more for methane, a lot more for nitrous oxide. In terms of Saskatchewan, it's even higher. So, we produce more CO2 of the total greenhouse gases in Saskatchewan come from agriculture than the other sectors. The methane's about the same. It's all based on, really, that's about cattle and enteric fermentation. And then for nitrous oxide, it's even higher. So almost 90% of all the nitrous oxide produced in Saskatchewan comes from here, and about 80% of that is from agricultural soils. So, and that's basically fertilizer use. And so basically what happens here, because of the global warming potential, what you've got is it takes about 3 1/3 kilograms of nitrous oxide to offset 1 ton of carbon that's removed from the atmosphere in terms of CO2. So we have to sort of, even though the emissions are generally small, they have a big effect in terms of global warming. So the real question now becomes, so what's the role for producers? Is there a way to sort of manage soils to sort of reduce some of that? And so our focus is really on nitrous oxide. And one thing that you have to remember is that nitrous oxide emissions are a natural result of processes. They occur through nitrification or denitrification, so whether it's aerobic or anaerobic, wet or dry conditions. So we're not going to get rid of them. We're not saying you're going to get zero emissions, because they just occur. They occur naturally, and they're always going to occur naturally. What we usually talk about is our emission factors. So that's the percentage of the nitrogen that's applied that's lost as nitrous oxide. What you can see, this is a graph that sort of shows about 1% is what the default characteristic is. So IPCC says 1% of the applied nitrogen comes off as nitrous oxide. But we know that that's not the case in Western Canada. It's an arid region. We don't get these big denitrification events. And so basically we're sort of in this range down in here, between here. About 0.4 to 0.6 is probably realistic and average for this region. And so our emissions are lower. But one of the things about this is that they consider all sources of nitrogen to be the same. So whether it's ammonium nitrate, urea fertilizer, whether it's cow manure, poultry manure, swine manure, or crop residues, nitrogen is nitrogen. And so if we're saying 0.4% of the nitrogen that you apply is lost as nitrous oxide, they say 0.4% of the nitrogen in crop residues is lost as nitrous oxide. And so the real question is that really the case? And so one of the ways to sort of look around this is we started looking at rotations. Can you sort of manage rotations to sort of manage some of this nitrogen input and output? And a lot of this was working with diversified rotations so that you're including pulses and oil seeds into basically cereal-based systems. The result is you get a diverse quantity and quality of the residues, so you're getting different types of residues coming in. This all has implications for nitrogen cycling or nutrient cycling in budgets. And also there's a big issue here about the fact that below ground nutrients aren't usually accounted for in doing this. We're not really looking at what's coming in through the root system, because it's really difficult to do in the field. And so just to give you an idea, this is just sort of a list of some of the research that's going on, looking at rotations by a variety of people here. So I'm not going to read through the list. You can sort of just take a look at it. But there's a lot of different rotation work going on using different systems. So pulse-based systems that have potatoes in them. There's working with perennialized systems, trying to identify mechanisms involved in all of this. We often see high emissions following canola, so not basically during the canola year, but during the subsequent year, we see high emissions on the canola residue. So that's other work we're going on. And then in the last two are these two studies. This is what I'll focus on for the rest of the talk. Is we have one study that's looking at quantifying the contribution of pulse residues to greenhouse gas emissions, nutrition, and growth of a subsequent wheat crop. And also there's some work that Reynolds has been doing looking at sort of the varying the frequency of pea and canola in rotations to see what its impact is. So the first study is really looking about what happens with these above and below ground inputs from post-crop residues. And so we've taken a slightly different approach. It's a field-based study, but we're using stable isotope labeling to sort of track everything. And so there's only a handful of studies like this, field studies like this in North America that have been conducted or are ongoing. And essentially what we're doing is we use nitrogen-15 and carbon-13 that we put in as labels to the plants, and that allows us to then follow that nitrogen through the system. So any sort of nitrogen that follows, any nitrogen-15 that shows up in the subsequent wheat crop has to have come from what we put in, and it's basically the nitrogen that's taken up by these residues and then is returned back into the soil. So we can track it all the way through the system. So part of this is also we want to find out really what's that below ground compartment. And so this involves, it's fairly time consuming. It takes a small army of summer students to do all of this. And so you have to recover the roots, so we have to recover a large mass. So we take big blocks of soil, basically about 30 centimeters by 30 centimeters by 60 centimeters deep. and then they're washed out. You recover all of the roots. Then you have to sort of go through all the water that you've washed and screen it and sieve it to try and recover all the rest of the smaller fine roots that wash out, and recover as much of the root material as you can from all of that. They're dried, they're weighed, they're analyzed, and we use all of that information to figure out how much total nitrogen is returned below ground. And part of the problem, this just goes to show you that these sort of root biomass changes over the course of the season. The anti-GAN from Ag Canada down in Swift Current had some studies where they were looking at this sort of root biomass, and you can just sort of see that over the course of a season, depending on where it is, down here, with lentil, not much early on, and then it comes up, stabilizes. Chickpea keeps growing, it'll just keep going all season long. Peas sort of have their maximum in the middle of the season, then roots start to die off, start at the end of the season. So it's hard to account for all of this, and that's where some of the N15 work comes in. And part of this is that we've got this below ground nitrogen, and the below ground nitrogen isn't just from the roots, it's also from root exudates. And so while these roots are growing, they produce chemicals that they just release into the environment, or small cells slough off, or small root hairs die off and they just fall off into the soil. And that's where all this nitrogen is coming from. And we had a study, there's a new professor at the U of S, Melissa Arcand, who as a PhD student looked at this and found that in some studies with lentils and canola, she could find as much as 50 to 60% of the total nitrogen below ground actually came from root exudates, not the root material itself. And so this is an important source, but it's hard to figure out what that is unless you're using labeling techniques. And even with that, we're sort of, we're getting a good estimate, but it's really sort of semi-quantitative. This research is all being conducted out at the Goodale Research Farm, about 15 miles from the university here. It involves a side-by-side comparison of pea, lentil, chickpea, fava bean, plus there's wheat. There's a non-fertilized and a fertilized wheat. Those are always in as our reference crops. The sort of continuous wheat with and without fertilizer is always our sort of baseline measurement. And it's all in rotation with wheat. So it was wheat, then the pulse, and then wheat is in the third year. So in 2014 and 2016, there were pulses growing on the plots, and then in 15 and 17, it's wheat. So we have one complete cycle. The 14-15 cycle is done, and the pulse cycle is finished for the 16-17 cycle, and we're currently in the wheat year of that second cycle. And the idea here basically is we have these plots, and within all of the main plot, there are subplots. One gets nitrogen-15 fertilizer, one doesn't get nitrogen-15 fertilizer. And when I say we're getting fertilizer, either they're pulse crops, we're giving them very small amounts. So it's the equivalent of 5 kilograms per hectare of an enriched N15 urea fertilizer. So they're not really getting a lot of fertilizer. They're still going to rely on fixation for most of their nitrogen, but it's just enough to get it into the plant so we can then follow it. And then, so you wind up with plants that have N15 in both the above and below ground compartments, and then we swap them. At harvest, we harvest all of the above ground material, we take subsamples, take them out, weigh them, analyze them, figure out how much is there, and then all of the above ground material gets moved to the plot that didn't have N15 fertilizer. All the above ground from that one goes to the plot that did. So now we have plots where the only N15 that comes up later has to have come from below ground material or from above ground material. And there are two other subplots within the main plot. One gets nitrogen-15 fertilizer and one gets nothing. So now in the wheat phase, we have essentially four small treatments within the large treatment. So for each crop, say for pea residue, there's now one where only the below ground material is labeled, one where only the above ground material is labeled, one where there's just an N15 fertilizer, and one where there's nothing. That lets us look at the background and the contribution from below ground residue, above ground residue, and fertilizer, and sort of put it all together to say where is the nitrogen ultimately coming from in the wheat crop and in the nitrous oxide emissions that come from there. So really, that's our objective here. We're going to compare these four pulse crops. And one of the sort of unique things about this is it's 4 pulse crops side by side under the same conditions, same field, everything is the same, so we can now sort of make comparisons instead of just saying, you know, here's a study where you did pea, or here's one with lentils, here's one with chickpea, and then trying to make comparisons when they weren't done at the same time or in the same place. This allows us to do all of that. We're looking at yield and uptake. We're looking at nitrogen fixation. We're measuring the contributions of the above and below ground to the next crop, and also the greenhouse gases associated with those different crops. And this is just an aerial photo of the site for during the pulse year, showing what it was. It's laid out. There's 24 plots out there. It's at harvest. It takes a small group of people to go out and collect everything, all the soil and plant samples that we need. This just shows it with the different pulses on there. And then so to get into what's going on, so the first question is, are these, the pulses are fixing nitrogen, so, you know, are they equivalent? What are they looking at? So here what we're seeing is, you know, you've got P, this is 2000, the dark bars are the 2014, the light was 2016, and you can see that faba bean fixes, you know, 85% of its nitrogen from nitrogen fixation. The chickpea and lentil are about 65 to 70%, and the pea is, you know, 55 to 60%. And it's fairly consistent across the years. And one thing about this, 2014 was a really wet year, and 2016, while it seemed like it rained constantly last year, the long-term average was only a few millimeters higher than the amount of precipitation we got during the growing season was only a few millimeters higher than the long-term average. And so we got a lot of small rainfall events, but no really big events. Whereas in 2014, we had some really big events, and we had about 50, I think it was about 45, 50% more total precipitation than we did, than we would normally get. So it's basically showing that the nitrogen fixation was relatively consistent across those times and across those different conditions. And so where's all this nitrogen going? We're also looking at seed yields, and you can see here, the big thing here is that P, while it's not fixing as much nitrogen as the others, is relatively resistant to change. It had, despite the fact that the conditions change, the yields don't change very much for P. Whereas you can see for chickpea in particular, chickpea was particularly sensitive to the environmental conditions, and under really wet conditions, the yields were fairly low, and when the conditions were a little bit better, the yields came up. Faba bean had high yields throughout. Also the same thing for the residue, and what you see here is if you're getting high seed yield, you get low residue yields, and part of this is always the nitrogen contribution of the pulses to the next year. And so, you know, if you're getting a good seed yield, which is what you really want, you're not putting as much residue back in, so you're not putting as much nitrogen back into the system. And so there's sort of that trade-off on where that nitrogen is coming and going. And then, so what's happening to all of that? So we looked at what's the source of the N in the harvested seed. So this top graph is 2014, and this is 2016. There'll be a series of graphs, and they're always going to be that way. The upper right is, upper left is the 2014, and the lower one is 2016. And so basically what you can see is that most of the N coming into the seed comes from nitrogen fixation. And in that sort of bad year 2014, you can see for chickpea, there's still most of it's from nitrogen fixation, but there's a fair proportion of that is actually coming from the soil. And in 2016, when conditions were better, you're still getting a fair amount from the soil, but the amount coming from nitrogen fixation is a little bit more. And so all of this N that's coming into the seed then is harvested and removed from the system with the seed. And you can see like faba bean fixes an awful lot of its nitrogen from the atmosphere, and then it turns right around and exports it all with the grain. So you're not putting in, on terms of the total amount, the proportion of N that remains in the residue is a lot lower for faba bean than it is for the other pulses. And then in the residues, this is sort of the same thing. We're seeing most of the N that's in the residues came from biological nitrogen fixation, and that's the same sort of thing. Differences across the years, but what you see here is that in the faba bean, the amount of N in the residue is a lot less because you've exported it in the grain. So now what we want to know is what is the greenhouse gas emissions. So we're looking at where's all of this going. In this, it's a relatively complicated table, but the bottom part in the red here is really what we're sort of looking at. That we do see differences between years in N2O emissions, and that's what we expect. It's really sort of weather driven and environment driven, and so you see a lot of interannual variations normally. And so we saw that, but the differences between crops didn't really change, and there's no real interaction between the weather and everything. And so they're fairly consistent results. And so what we're seeing really is that down here, non-fertilized wheat had low emissions. That's what we expect. The fertilized wheat had the highest ones, but the pulses had a high one, and it was almost as high, not, sort of intermediate, but not really all that different from the fertilized wheat. And the thing that's not shown in this graph is that this is the total emission. So our end budget year goes from when the fertilizer goes on till just before fertilizer goes on the following year. So it's the growing season into the late fall, and then the spring thaw the following year before the next crop goes on. And if we looked at just growing season emissions, the pulse crop looks pretty much exactly like the non-fertilized wheat. The difference is the pulse crop residues break down pretty easily. They've got a lot of nitrogen in them. When they break down and decompose, they release carbon that the microorganisms need to convert nitrogen into nitrous oxide, and then they freeze over the winter. So now you've got a little frozen soil that has a readily available carbon source, it's got a readily available nitrogen source, and come spring thaw, after the snow starts to melt, it's got a readily available water source, and you basically have engineered the conditions for denitrification to occur, and you get a big pulse of nitrous oxide that comes off, and that's what shows up here, is all of a sudden the emissions on this one jump from being relatively low and equal to a non-fertilized wheat crop to something that looks more like a fertilized one because of that big pulse of nitrous oxide in the spring. And there are some differences there, and that'll sort of become a little more apparent as I sort of move through some of the rest of this. So if we look at the source of the N2O emissions, you can see there are differences. The soil as fertilizer still takes up a big part of this. If you look at this is sort of if we don't consider the fertilizer and just look at the residue in the soil, what you're seeing is the soil nitrogen. So soil organic matter only contributes about 3% of the N2O that comes off. And so that's the organic matter that was there when we started all of this. About 20% of the nitrous oxide that comes off came from the above ground residues. And about 77% comes from the below ground residues. So in terms of the residues, everything that's below ground is the really important part here. But if we look at the fertilizer, so now we've added in that fertilizer component, you still go back and see fertilizer is the overwhelming sort of source of nitrogen that winds up as coming out as nitrous oxide here. That now, in terms of the total emissions where everything is coming from, 90% of it comes from the fertilizer, 10% from the residues, and really a very minimal, almost tenths of a percent from the soil itself. So the residues have a component here, but they're certainly not contributing an equal amount to what the fertilizer is. So we can go back now and we can confidently say here that the emission factors associated with the residues are much lower than they are for fertilizer. And so when it goes into an accounting form of how much nitrogen, when you go back and say you're going to, if you have to account for all of this nitrogen, you can go back and say, well, if a kilogram of nitrogen added as residue is not the same as a kilogram added as fertilizer. It might be about the same as 2/10 or a quarter of a kilogram of fertilizer nitrogen. So that's where all of this comes in, is in that sort of eventual accounting part, and also just in the fact that you're getting low emissions from these pulse systems. So in the wheat year, 2015, we have one complete year for wheat, and so that was 2015. 2015, while 2014 was very wet, 2015 was very dry, and we actually had to take water trucks out there to keep the crops alive, basically. It's an experiment that was with the, one thing about stable isotopes is they're stable, they're not radioactive, they're easy to work with, and they're expensive as all hell. And so this is an expensive study, and so in order, when the fact that we had drought conditions, we needed to sort of bring water out to the site to keep everything alive to maintain our investment in this and into the investment of the Pulse Growers Association. So what we looked at first was after we took the pulses off, we went in the fall, we do soil tests. We wanted to know how much inorganic nitrogen is left behind. And you can see from here that peas and lentils leave the most in both years of the both of the pulse years. We're getting the most here, less amounts with the chickpea and fava bean. The differences here and here are basically, again, due to the weather effects. But really, pea doesn't fix a lot of nitrogen, but it leaves a lot of nitrogen behind. The lentil is sort of intermediate in terms of fixation, but it has the quality of the residue is such that it breaks down pretty easily, and so it leaves a lot of nitrogen behind as well. So then if we look at the wheat, we want to know, okay, so where's all the nitrogen coming from? So we look at yields, and these are the yields of the wheat grown on the stubble. So that's on pea stubble, lentil, chickpea, and fava bean stubble. And through all of this, because there's different amounts of nitrogen left in the soil tests came back and said, okay, for pea, we added about 50 kilograms of nitrogen fertilizer to the wheat in the spring. For fava bean, we added about 65, based on the soil tests for the individual crops going into it. So essentially, between the amount of N that was there in the residue plus the fertilizer N, they should be roughly equivalent, have the same amount of N going into it. And despite that, the wheat yields on pea and lentil are significantly higher than they were on chickpea and fava bean. And the same thing holds for the nitrogen. So how much nitrogen is recovered in the wheat, we're still seeing that there's a lot more of it on the pea and lentil. And so in terms of putting in a nitrogen credit to the next crop, going into this, everybody was thinking, well, fava bean fixes so much nitrogen, it's got to leave all this nitrogen behind, it's going to have a really high nitrogen credit. It actually looks like fava bean has the lower nitrogen credit of all of them. And the chickpea is just so sensitive to weather that its contribution sort of varies. If it's a good year, you get a decent one, and if it's not, you don't. So then where's all this N coming from? We go back and we look at this, and we can break it down, and you can see that most of the N in the seed and most of the N in the residue or the straw is coming from below ground sources. So when we partition everything out, you've got above ground residue going back in, you've got below ground residue going back in, and that below ground residue provides much more nitrogen than the above ground residue does to the following crop. And one thing that I should say on this is that this was in 2015. We actually grew barley on this in 2016, and we followed it again, and we saw the same thing. So there's nitrogen carrying over from the pulse residues 2 years past when the pulse was harvested. And we have oats on it this year, and we'll follow it this year into the oat crop to see what's going on. So we had about 27% recovery of the residue N in the wheat, all told, across all of the crops in the first year, and we had about 17% recovery of residue N in the second year's crop. So it's not just a one-year thing, it carries on for multiple years. So basically just sort of, we're still wrapping this up. We have carbon data, we have carbon dioxide data. The carbon dioxide data is different from the nitrous oxide data in that most of the carbon dioxide that's produced through through microbial respiration, soil respiration, actually comes from the above ground residue. And so it's probably 4 times as much from the above ground residue as the below ground residue. So while the below ground is contributing nitrogen for nitrous oxide, the above ground residue seems to break down a little more and gives us CO2, but not a lot of nitrogen. And so we're sort of looking a little closer at that this year. So essentially during the pulse year, what we found, biological nitrogen fixation, fava beans greater than lentil, chickpea or intermediate, pea was the lowest. but the chickpea seems to be the most sensitive to weather patterns. And from in terms of the above ground residue yields, chickpea was greater than the lentil and the pea and the fava bean, but that again doesn't necessarily relate into a big N credit for this crop. And in the wheat year, there's more N in the soil from the lentil and pea residue than there is from chickpea and fava bean. The wheat yields are higher on the pea and the lentil than they are on the chickpea and fava bean, and that the source of N in the seeds and the straws is mainly from the below ground. And it's not saying that that's all where it's coming from, because there's still a large contribution from fertilizer that year, but in terms of residue contributions, the below ground residue is the more important. And I think from this study, that's sort of our take-home thing, is that below ground nitrogen and that root compartment is really important. It's really more important than we've given it credit for in the past, and it's really difficult to get at. And I think this afternoon in the panel with other people talking about roots and phenotyping, you'll probably hear more about that. But this is where we need to do some work, is to find better accounting for what's going on below ground for these crops. The second study is one that Reynolds been leading, looking at the frequency of pea and canola in rotations. And it's basically, when they talk about using canola for bio-oil or biofuels, there's a tendency to want to get it into rotation more frequently, and the same thing for pea. Pulses are becoming more popular, and so it's the same thing. What happens if you put them a little more frequently into rotation? And so this is research that was done out at the Scott Research Farm, and the treatments here are a wheat plus nitrogen, peas. So pea is just continuous pea. They just grew it for 4 years or so. And then a pea wheat rotation, canola wheat, and pea canola wheat. And in each year of the study, all phases were present so that they could sort of do the greenhouse gas accounting across all of that. And so this is just jumping right into some of the greenhouse gas data. And so this is the wheat. And so here's what they're doing here was looking at yield scaled emissions. So those are N2O emissions on a yield basis. So we're trying to sort of go in and say, okay, part of this is if you put on more nitrogen and maybe you get a little higher nitrous oxide emission, but you also get higher yields, so is the intensity of it, so is the amount of N2O produced per unit of yield higher or lower than it would be in the other system? So that's what this one was looking at. And so what you can see is that it has really high emissions. So there's this is the 3 years cumulative. So over the 3 years of the rotation, 3 consecutive years of wheat, this is what was there. So we get these really, really high emissions, and they're the highest of all of them. And then if you come in here, this is, you start seeing lower ones. Continuous pea has wheat on pea has a lower emission. Wheat on wheat has a lower emission. It's the wheat on canola is high. And then, from a rotation basis, looking at everything, the wheat on canola again is high. And we've seen this in a few other studies, and we've done some greenhouse studies that show the same thing. And we also see the same thing with flax, which was really surprising, because flax, the amount of decomposition of the flax is much lower than it is for any of the other crops, but we still see high emissions for like wheat on flax than we normally do. So there's something about oil seeds that seems to be pushing emissions in there. So there's what we believe is some biochemical factor. It's not really related to the amount of nitrogen there, because the amount of nitrogen in, say, the canola that was returned with canola and the amount of nitrogen returned with pea was very similar, but the emissions are higher on the canola residue. And so we've been talking to Martin Rennie in the Plant Sciences Department, who does a lot of work on the biochemistry of canola oils and things, and they think there's a biochemical mechanism for it. And Bobby Helgeson, who's a research scientist with Agriculture and Agri-Food Canada here, a soil microbiologist in Saskatoon, has said that she's seen some papers that say that some of the materials in the canola residue are either turning on the genes that cause nitrification or denitrification to occur by microorganisms, or it's turning off the genes that prevent that would take nitrous oxide all the way down to nitrogen, so it sort of builds up a little. So we have a study that's just beginning to look at all of this now. But essentially, crop sequence and crop type are influenced N2O emissions on an area basis and on a yield scale basis, that including canola in the crop sequence costs the overall rotation on both a yield and area scaled one, and that including a pulse benefits the overall rotation on the same basis. And so when you look at a rotation that contains all three crops, it's really not so bad, because you're getting a little extra after the canola and a little less during the pulse year, and so they sort of balance off on a rotation basis. So it's part of it is, in the past we've always looked at crops individually. You look at peas, or you look at wheat, or you look at canola, and it's basically because the funding comes from the Wheat Development Commission or the Canola Development Commission or SAS Pulse, and you're looking at that crop. Now that we start looking at whole rotations, you can still see, you can go back and say, okay, yes, there is a little cost associated with canola, but if it's included in a rotation that includes a pulse, you can sort of negate that cost on a rotation basis and sort of pick up what's going on. And so part of this is, this is the other side to the carbon sequestration. We're not looking at carbon sequestration in these because, as Brian talked to you earlier, looking for small changes against a large background in a short time interval of 2 or 3 years just isn't possible. And so in order to do that, we would need to be looking at things a little more long term. And that was one of the things I was asked, was sort of do we have knowledge gaps? Where would we like to see things going? And Reynolds and Diane and Bobby and I have all talked about this. And I'm not going to read all of it, but we do see gaps in things. One of them is that we do need rotation work, that you need this integrated work that looks across them. And really, if we wanted to do this right, we'd be saying we really would like to carry these studies out for maybe 2 full rotations, so maybe 6 years. And that's the problem. One of the gaps here is that it's really difficult to find anybody who'll fund you for 6 years on a project like this. And so I just want to acknowledge all of the people who sort of have helped fund all of this. Sask Ag Ministry through the Agriculture Development Fund and the Strategic Research Program in Soils and Environment, Agriculture and Agri-Food Canada, SAS Pulse, Western Grains, and of course, as Brian and Daniel and everybody else has alluded to, it takes a small army of people, and these are the people involved this year. And so every year there's about that many, but all the people on the right-hand side change every year. Those are the summer students. So, and thank you.